The title of my research it's the role of LabBA0473 in osmotic pressure in Leptospira biflexa. LabBA0473 it's a gene that we find that relates to the bacteria survival in water. The pathogenic strain of this bacteria, it's the most generous or worldwide spread zoonosis in the world. It affects 500,000 people annually, from which there are more than 25% deaths. It has a lot of importance for the public health.
We do Leptospira research. The reason we do that is because of a disease that's found all over the world called leptospirosis. There's over 500,000 cases of severe cases of leptospirosis annually. We study a non-pathogenic strain, so it's very useful to use as a pedagogical tool with our students so they can use it without fear of becoming infected themselves.
Coming into my junior year, I was studying in theory, I was studying biochemistry, principles of microbiology, principles of genetics. And now, coming into my senior year, I'm able to put all of this knowledge into practice. So far we're working with 2 specific mutants.
We've created a random transposal mutant library. We've got about 1,000 mutants completed to date, so that's about 20% of the library we hope to create, and at some point also disseminate to other researchers. Where we're moving next is, one of the really exciting things is that we have collaboration set up with major institutions in France and also within the northeastern US. With those collaborations, one of the interesting things is that we're going to be able to do some of our studies here and other institutions as well and find out even more information. For example, if the pathogenic strains have similar effects as what we've seen in the non-pathogenic strains. It was an absolute pleasure to work with Pauline on this project. I remember the first time Pauline found me, I was walking through the hallway and she came around and asked me, Dr. Pappas, I'm really excited about your research program. She's one of these students that's really ambitious, a real go-getter. She started her project early, which is always encouraged. She's been working on the project for over 1.5 years now. I'm really excited to see what she's going to be able to do next when she leaves Manhattanville as well, with what she's gained being here.
Working with Dr. Pappas has been fantastic too. He's allowed me not only to see him, but also to try it myself. Sometimes researchers are not so open because materials and media, are difficult to find, are expensive to produce. It's been fantastic how openly he received me in the lab, and he showed me so much.
So the college has really dedicated resources to students and their research projects into the science program at Manhattanville.
I applied to graduate programs. I intend to proceed with a graduate study, degree in biochemistry and molecular cell biology. This experience has been very, very rewarding. Everything that I learned can be applied. Everything that I learned will be applied.